specializing in therapies directed at the G protein receptors. I think, Rabbi, if you can mute everybody's right now, that would be great. So anyway, the bottom line is Mark is a very, very smart guy who knows a lot about pharmaceuticals. And he's in his current position with Sorrento Pharmaceuticals. We will share the latest developments in the diagnosis and treatment of COVID-related diseases. But in keeping with the usual format of this event, we will start with a word of Torah from Rabbi Reich, addressing once again the importance that health plays from a Torah perspective in our lives. So, Rabbi?
Thanks, Brian. And it's a great honor that I get to be the lead-up batter to Dr. Brunswick, who could probably give the Torah presentation as well as the science presentation this morning. But I'll just share a few notes that are important to keep in mind that I think are going to help frame the critical nature of Mark's work. You know, the Chassam Sofer in the 18th century, early 19th century, was for the Oberlander region of Slovakia, Hungary, was the Jewish leader, the communal leader amongst Orthodox Jewry. And he, at the time, was really under the gun and being pressured by the Enlightenment movement. The Enlightenment movement, Reform Judaism, and the like. And it was basically, he considered it like his life's mission to do whatever he can to make sure that orthodoxy was sustained, and that Torah was the prime activity for the Jewish people, and that it did not waver in its observance of Torah mitzvahs in even in the slightest fashion. So he was posed a question which, based on his whole modus operandi, you would be surprised by his answer. The question was, when you have a conflict between Torah and science, so what is one supposed to do? So if you have, let's say, let's say an example for something like this would be the Gemara has in Masechah Shabbos that a good way to cure pimples is by washing your face in beet juice. Okay, so I haven't spoken to a dermatologist recently, but all of my friends who had acne, I didn't see any of them whip out a bottle of beet juice and start washing their face in beet juice. I can't imagine that science today would agree to such a thing. So, but on the other hand, the Gemara tells us, the Talmud tells us, that's what one's supposed to do when they have pimples. So what is one supposed to do when you have a conflict between our Chazal, between our rabbis, between Torah and science, modern science on the other end? So the Chassam Sofer, if I were to guess what his answer was going to be, I would have guessed he would have said, well, we have to hold on to our traditions. There's nothing greater than our Torah Kadosha. There's Ruach HaKodesh. In fact, this is an approach that is taken by the Ravid. The Ravid is quoted in an amazing sefer called the Shem HaGdolim by the Chida, which was a Sephardic contemporary of the Chassam Sofer. The Chida, Chacham Yosef David Azulai, was a Sephardic contemporary of the Chassam Sofer, where he has a beautiful work of biographies of Rishonim and Achronim. And what he quotes when he has his entry on who the Ravid was, he says that, you know, the Ravid felt that there was a certain Ruach HaKodesh, a divine inspiration to know not just Torah, but know even science and secular knowledge by our great rabbis, and therefore you can't deviate from the words one iota even in these realms. So that's what I would have assumed the Chassam Sofer would have said, especially considering the fact that he was up against a modern turn of the world of enlightenment and reformed Judaism and the like. But that's not what the Chassam Sofer says. The Chassam Sofer says that God put into the Bria, God put into creation, into nature, how things are supposed to work. And that's exactly what Shlomo HaMelech tells us. Shlomo HaMelech says, Ein kol chadash tachas hashemesh, that there's nothing new under the sun, that all of the raw materials all the raw materials needed in order to, let's say, create an iPhone already were in existence by the time of creation. It's just a matter of development and figuring out different things. And eventually what you have is, thousands of years later, you have an iPhone. And the same is true with all science, all medicine. There is discovery, there is learning, there is developing, but there's nothing new under the sun. That doesn't mean, says Rashi, that whatever existed at the time of the creation of the world is the way that things need to be done. Whatever existed at the time of Har Sinai is the way that things need to be done. When our rabbis, when they said in the Gemara that the best way to get rid of pimples is by washing your face in beet juice, you know how they figured that out? It's because they went to the local doctors and they asked the local doctors, what's the best way to get rid of beet juice? What's the best way to get rid of pimples? By washing your face in beet juice. That is what they went to the scientists of the time to figure out what to do, because we have an obligation to take care of our health. We look at the world around us to figure out how to best take care of our health. And we are mandated by the Torah, says the Chassam Sofer, to look at our doctors, to look at our scientists, to figure out what to do. I think that this has become all the more central over the last year as we've been dealing with this virus, recognizing just how critical this dual relationship is, of knowing how to look to outer resources to be able to take care of our health and to be able to take care of the health of others and the greater good, because that is exactly what the Torah mandates of us to do. And it's our great honor and pleasure that we have in our midst, that we have a doctor in our community who's really on the front lines of trying to figure this thing out, to squash this virus and to squash this pandemic, and to be fulfilling this mandate from the Torah of Rapo Yirapeh, that we look at the scientists and doctors to be able to solve our medical, to be a shliach, to be a agent of God to solve our medical issues, and we fulfilling the Chassam Sofer's mandate of that when there is an issue, you need to be able to go to the doctors to be able to figure out the best solution. So now we turn to our resident doctor. We turn to Dr. Brunswick to figure out, what are we going to do if, God forbid, somebody gets COVID? What's the latest developments? We're going to hear from Dr. Brunswick now.
Okay.
So Mark, just before you begin, I want to just ask all the participants here if there is, I'm sure there'll be questions and answers at the end of the session, but if you want to text by using the little box down at the bottom, the chat box on your screen, why don't you just send your questions via chat, and Mark will try to get to them all at the end of his presentation. I'll turn it over to you. Thank you, Mark.
Thank you, Brian. So what I'm gonna do is I'm going to go through my corporate presentation. And the reason I'm gonna do that is because since I'm a senior guy at my corporation, if I go off track, the SEC might come down on me. And I really don't want to deal with the SEC. So I'm going to use my corporate presentation to keep me on track. I will talk in generalities about some of the other products that are not mine, but I won't go into specifics because I don't want to bash another company or not bash another company. I'm going to try and share my screen so you can see this presentation. Let me know if it works. Can you see it?
Yes.
Yeah, very good.
I'll see if I can make this full screen. That should be better. So as you can see, this was a presentation that we put together in January of this year, so not so long ago. this is to keep the SEC happy. What was, what is, and what will be, who knows. So Sorrento was founded in 2009 by Henry G, our CEO. The moment we've got a little less than 500 employees, with over 100 employees with PhDs and MDs. We've got over 300,000 square feet in both research and CGMP. CGMP means that you can manufacture drugs to be given to people. And the GMP means good manufacturing practices, and the little c in the front of the CGMP means current. And what that means is that if somebody manufactures something using methods from the 1940s, that would not be good now, because they don't comply with the C, the current. And so as part of my job, I have to ensure that all of the manufacturing that we do in-house is maintained up to the current standards. We've got 5 CGMP facilities, 4 in the US and 1 in China.
Mark, I think we're having a little bit of trouble seeing the PowerPoint presentation.
It says, is that better now?
Yeah.
Okay. So we've got 5 CGMP facilities, 4 in the US and 1 in China, where we make things like small molecules, which are what are called regular drugs. We make antibodies, we make cell therapies. We also have a facility in Atlanta where we make a device that we can inject either small molecules or antibodies just under the surface of the skin. And the purpose of that is if it goes just under the surface of the skin, the drug is delivered to what's called the lymphatic system, as opposed into the intravenous system. And the advantage of delivering some drugs, not all drugs, but some drugs into the lymphatic system is that the volume of the lymphatic system is about 1/10 of the volume of the blood system. So you get an effective dose, you can be 1/10 of the amount that you give. And if you're 1/10 of the amount of the dose, then you would also be much lower in adverse events. So we're investigating that, and we're having some good success with that. We've got 1 approved FDA drug, which is called ZT-Lido. And what that is it's a lidocaine topical system. And the advantage of this particular drug over other lidocaine topical systems, which are, you can see them in CVS and other places where it's a lidocaine on a adhesive device that you stick on where it hurts, and then it relieves the pain. The other devices, they tend to fall off, and you can't exercise in them, and you need, in order to keep them adhered to the skin, you need to use plasters or sticky tape to keep it adhered to the skin. This particular drug and system will stay attached to the skin very well. One of the CEO of the company we purchased that had it, he used to, in demonstrations, he used to put it on his hand and move his fingers for half an hour while the giving the presentation, and the material stayed on his hand. So this is an overview of Sorrento, and if you can see in this bottom corner, it says in last year we submitted 12 INDs. 12 INDs for a small company like us is a huge number of programs. As you can see from this wheel, we've got drugs in everything that you can think of that are on the cutting edge of all of the pharmaceutical industry. This is a very high level of what we do, and the important thing is the foundation, which is over here. which is called Gmap. What our CEO did when he first started the company was he took 600 individuals, both males and females, across the various races. And from those people, he isolated their B lymphocytes. B lymphocytes are the cells that make antibodies. So he had a large number of cells from all of those people, and he isolated the antibodies from those people. He then basically took an antibody. If you look at this particular picture over here, can you see where I'm moving the mouse? So you can see that there are 4 chains. There are 2 little red ones and 2 large red ones. Those are called the heavy and light chains. The large ones are identical to each other, and the small ones are identical to each other. And that's a depiction of an antibody. What he did was that's the antibody that he isolated from the patients or the donors. You can separate out molecularly the light chains and the heavy chains, and then he threw them in the air and let them reassemble how they wanted. And what ended up happening is we have a library of 2 × 10⁻XVI independent monoclonal antibodies, and these are all fully human because they're all isolated from humans. 2 × 10⁻XVI is a 2 with 16 zeros after it, which is a pretty large number. And that library, if not the largest, it's one of the top 3 libraries in the world. And from that library, we can pick out an antibody that is directed against any antigen of interest. An antigen is something, a protein on the surface of a cell, or the protein on a virus, or a protein on a bacteria that you want to have an antibody against. Before COVID, we were using this library to generate antibodies against tumor cells. And it's like fishing. You take these antibodies, you put them in a pot, you dangle over your antigen, and then you find antibodies that bind to it. And then you find for the viruses or for tumors, you put it in, you fish out the antibody, and then you go ahead and find the antibody that's really the most effective. When COVID came along, we did that, and we took the COVID virus and we put it into our pool of antibodies. We used, I think it was about 1 billion independent antibodies. And from that pool, we selected 500 antibodies that seemed to bind to the virus. Upon further analysis of those 500, that got whittled down to about 30 to 40 antibodies that bound quite well. And then we investigated that binding further, and we isolated one antibody that not only bound very well, but it also completely inactivated the virus. And by inactivating the virus, that meant it interfered with the way the virus interacts with the cell to infect it. So it prevented infection of the cell. That antibody originally was called, what we called it was Kovi Guard, and it's also known as STI 1499. That antibody was developed in the clinic, and we have treated healthy volunteers with that antibody. But while that was being investigated, we also did what the body normally does in an immune response, and that's called affinity mature the antibody. So when the body makes the initial response to an infection, it does its best shot at how the body can defeat the infection. But as the infection progresses, the body refines the molecules that it's using and makes them better. That's called affinity maturation. And so we did that in the lab, and we changed 1499 into Kovi AMG, also known as STI 2020. And what that did was that took an antibody that was already very, very potent and made it about 100 times more potent. As a reference point, in studies, we've shown that an effective dose in humans of this antibody, and we're investigating that now, should be of the order of 40 to 100 milligrams of antibody would be effective at fully neutralizing COVID in patients. Currently there are two companies that have approved antibodies using what's called an emergency use authorization from the FDA. One is Lilly, and their antibody is given at 700 milligrams. So we're about a 1/7 to a 14th of the amount of antibody that they give. And the other company is Regeneron. They have a mixture of two antibodies, and they give between 2 and 8, actually it's 2.8 milligram, 2.8 grams and 7 and 8 grams. So we are about 1,000-fold better than them. The reason that's worth pointing out is because the amount of antibody you can make is pretty fixed according to the way we make antibodies in culture. Antibodies are made in a fermenter, which is pretty much like making beer. You go, if anyone's been to a brewery, they've seen these large stainless steel containers that you make beer in. They come in various different sizes. We have a fermenter that's 2,000 liters. The fermenters go up to about 30,000 liters. And when you make an antibody, you can get between 1 and 5 grams of antibody per liter of material, of the container that you're using. So if you're using a 2,000 liter reactor like we are, and you have an antibody that gives 1 gram per liter, that gives you 2,000 grams per production run. And then that then follows into how much antibody you can use to treat patients. So if you're only giving 40 milligrams, you get a lot more bang for your buck per run of fermenter than if you have to give each patient 8 grams.
How long is the fermenter process, Mark? So time-wise?
So time-wise, from thawing the cells till the end of production is about 2 months.
Oh.
And at any time, if somebody sticks their finger into something where they shouldn't have stuck their finger, that's the end of that production run, and you have started from scratch. So here's this overview of Sorrento. We have the non-opioid pain management, and if we get time, I can talk about that later. And then we have immuno-oncology, and I can talk about that later as well. But what we're all here to hear is this middle bit, this COVID-19. And we started on COVID-19, as did everyone else, at the beginning of last year. So we've been at it for nearly a year.
Mark, there is one question here that might be appropriate while we're talking about the antibodies, and that is, with the new variants, is it still effective, or does that send you back to square one? So
one of the reasons we stopped with 1499 was it stopped being effective against one of the variants. 2020, the AMG, Kovi AMG, is still effective against most of the variants. Some of the variants are escaping, but we have antibodies that are active against that who are coming along right now. And one of the things we've spoken about in the past is what Regeneron are doing, are using mixtures of antibodies. So you would have 2 antibodies put together to cover the variants. But the virus is varying very, very fast. So the point, it's always a catch-up game with the virus. But if you've got an antibody that interferes with the way the virus interacts with the cell, I don't know if you can see my hand hitting my fist. If you can put something in between the fist and the hand, that'll prevent the infection. And that's what these antibodies are doing. Whether they bind to the side of the virus, so sort of semi-interfere, or they completely interfere, that's the difference between the various antibodies. But we are working on, our current antibodies work on everything except the South African virus.
Ah.
South Africans, yes. They're always the problem. So our philosophy is to detect early, then treat, and then monitor infection, and people go on their merry way. So in this cartoon, what we've got is a very simple test that we can do that has been shown to be about as equivalent as what's called the PCR, which is polymerase chain reaction, which is the gold standard. The problem with the gold standard is it takes a good number of hours to get a result. You know, I was in a meeting where somebody, after we'd been in an all-day meeting, they suddenly said the next day that they've now been diagnosed with COVID. And so we all had to go and get tested. So I went to the rapid COVID testing it was in Carlsbad. They charged me nearly $500 to give me a result in 2 hours. I got the result 12 hours later. So that's one of the problems. You know, someone else also went there and they lost his sample. And they didn't tell him about it till the next day. So what we have developed is take the PCR takes a swab and puts it way, way up in your nose. It's almost like a brain biopsy. Our swab takes just a shallow nasal swab. That's just almost just like blowing your nose. Just rub the swab around the nose, and then you extract the virus, then put it on this cassette, and then in 15 minutes, you can see the result, either negative, that's just one line at the top, or positive when you have 2 lines. You can then have telemedicine if you want, and then we have 2 options for treating you at that point. You can get Kovi drops, which is STI 2020 formulated as a dropper. So you put some drops in your nose, because the nose is what you just diagnosed as where the virus is. And that will prevent any further infections going down to your lungs, and will also prevent you breathing virus onto anyone else. We would also recommend at that point getting Kovi AMG, STI 2020, as an IV push. So because we're so potent, we can give our antibody in about 15 minutes. I'm sorry, in 5 minutes, as opposed to 2 hours, which is what Regeneron and Lilly are doing. One of the reasons that you want to try and give it very quickly is because then there's likely to be less side effects. And also, you don't have to stay afterwards for 2 hours in the hospital. With Regeneron and Lilly, their infusion is over 2 hours, and then you have to stay 2 hours after the infusion. So it's a half a day process to get the antibodies. And then after you've been treated, the goal is to then go from 2 lines over here to only 1 line. So we have a whole range of treatments going from prevention to diagnosis over here, and then treatment at various stages of the disease. I'll start with this bit here. This is Kovi gene mAb, and what this is we've got a grant from the Department of it's called DARPA, it's a Defense Agency grant, that we've taken the gene for the antibody and we're putting it into what's called an expression system, which means that you can take the gene and you put an injection into somebody's arm, and their arm will act like the fermenter. And they will be able to produce as much antibody as they need to prevent an infection. The gene, once it's injected, will produce antibody for probably 6 months, and that would give us 6 months protection. If we find that we need to put in another antibody because this original antibody is no good, we can just use the other arm and put in, on one arm you put in one antibody, on the other arm you put the other antibody, and then that patient would be fully covered for all infections.
How does that differ, Mark, from the vaccines that are being offered now, or is it similar?
No, so I'm glad you asked that question. A vaccine relies on the body to make the right choices when it makes the immune response. Sometimes the body can't make those choices, i.e. the patient is immunocompromised. An immunocompromised patient can't make an immune response, and therefore you can't give them a vaccine because the vaccine wouldn't do anything. Elderly patients are somewhat immunocompromised, so they have a harder time making the immune response. Also, you can't ensure that the immune response is the correct immune response. The body can sometimes make a mistake and make an antibody that would be detrimental to the patient. And there are two ways that antibody could be detrimental. One is called antibody-dependent enhancement. So what that means is that the antibody, instead of inactivating the virus, the antibody takes the virus and helps it infect cells that it wouldn't have infected in the first place. And when that happens, those cells can get depleted, and that can lead to all sorts of bad outcomes. That hasn't yet been seen with COVID, but it has been seen in other viruses, particularly RSV, which is a children's virus. Some of their immune responses with that sort of wipe out the immunity of these babies. And so that's something that has to be watched. What we did was when we made our antibodies, we edited out the ability of these antibodies to be able to do that. So our antibodies cannot cause ADE. The other thing that can happen in an immune response is that the immune response can react against the virus, but also against a self protein. So what that potentially means is that, and this I know to be a fact, that some of the sequences of the virus are shared with normal proteins in humans. One of the proteins that is known to share sequences is a protein called interferon gamma, which is used as the body's defense against viruses. So you've now got the virus enforcing, or not enforcing, helping the body make an antibody response against the normal protein, and that's somewhat of an autoimmune disease. There are also reports, and it could just be social media type reports, but often these have some basis in fact, that the virus shares sequences with antigens or proteins present on the placenta, which potentially, if that is true, could mean that a woman who is pregnant or wants to get pregnant could have a problem with a pregnancy if they've been vaccinated. So,
Mark, there is one other question on the subject. While Nora Lakin wants to know, how does the gene MAP get into the cells?
So it's injected, and the membranes. It's in a micelle, which is like an oil-based concoction, and when it hits the cell membrane, it combines with the cell membrane and fuses with the cell, and then it just goes in, and then it sits. It does not integrate with the patient's own DNA. It sits outside the patient's DNA, but it sits in the extracellular, in the domain inside the cell. It will then get transcribed by the cell's mechanism of transcribing proteins. So then moving along, we've got 3 different ways to diagnose COVID infections or responses to an infection. Kovi Trace, this is a technology we licensed from Columbia University, and that's currently being examined by the FDA under an emergency use authorization. This is a saliva test that's very sensitive and will give a result within about 15 to 30 minutes, sorry. And we've further developed this to avoid people having to spit and contaminate people around them, to being a nose swab test. Then there's Kovi Stix, which I spoke about, and that's also under EUA examination by the FDA, and they've had that since the end of last year. And when people ask me when are we going to hear, the only response I can give is whenever the FDA tells us. It's there. The FDA has no timelines for any of these reviews. Unlike reviews for drug applications where there are strict timelines, there are no timelines for these types of reviews. And then we have Kovi Track, which we had an early version of, that we've now further developed to be much more sensitive to detect antibody responses, both IgG, which is the routine response, and IgM, which is an early response. And we've used that to monitor people within the company who have opted to have vaccinations. And so there are a number of people who have, and so we monitor their antibody responses in-house. Then these 4 things on the right-hand side are our treatment options. So I've already spoken about 2099, which is the drops, and the IV push. Those are for early, either as outpatients or early hospitalizations. SCI 5656 is a small molecule that has been examined in over 600 patients for non-small cell lung cancer, and it appears to be quite an effective drug. What we're using this for in COVID is that one of the side effects of late-term disease is called cytokine storm. Cytokines are immune reactive proteins that the body produces to enhance the immune system and get things going. But what happens with COVID is the body goes overboard and produces too much interferon, too much interleukin 6, too much TNF alpha, and the body just starts reacting against itself, and then patients go rapidly downhill from there. So what this drug does is it's very effective at controlling that cytokine storm. So when patients are in the ICU, once they get there, chances are they are in cytokine storm, because otherwise they wouldn't need to go into the ICU. And this drug can be given to control that. We've got this trial going on currently in the US and in Brazil. And I can't tell you any results because the trials are what's called blinded trials. What that means is the half the patients are getting drug and half the patients are getting placebo. And so until the study is finished, which we hope will be soon, but we're approaching the endpoint of the US trial of the number of patients, and in Brazil we're getting up there in the numbers of patients, and we should be able to have some results out probably in 2 months or so, and we will know whether this drug is having an effect. Then the last one over here, STI 8282, which are mesenchymal stem cells. These are cells that were isolated from a donor who was having some liposuction. They had some liposuction, and then stem cells, which are known as pluripotent cells. But what that means is these cells can differentiate into anything that the body tells it to. So these were isolated from this donor, and patients, ICU patients, just about to be intubated or newly intubated, are given these cells. And we've had a press release on this. The first 4 patients who were treated with this therapy, so all at a single center in Fresno. The therapy is given on day 1, 3, and 5. And all of the patients were discharged from the hospital, not just from the ICU, from the hospital by day 5. So this is quite a remarkable therapy that, if it pans out, at the moment it's only in 1 center. We're expanding. We hope to fit with this trial should finish soon, and we will meet with the FDA to discuss how this is going to go forward. But the way it's looking now, this is something that would be very advantageous to anyone who is newly into the ICU. And because, as I said, these patients are walking out and going home in less than a week. And in general, patients, once they get into the ICU, there's somewhere between a 20 and 50% death rate. So going into more specifics on Kovi Stix, do people want me to get into more science, or?
I think, Mark, there are a few questions here. We want to finish up in 15 minutes if you want to go to the questions.
I'll take questions. I've done a good overview of what we have.
Great. And so if you want to go to your chat there on the chat, or I can just read them out to you.
Okay. So the first one at the top.
You've answered the first and second one. Then Miriam Geller, can you please talk later about the vaccines, Moderna and Pfizer, that people are getting now?
Okay. So the Johnson & Johnson vaccine is using, what's called an old technology, which is that there are ways to express antigens in people in order to induce an immune response for a vaccine. And what that is you take a virus and then you add the genes for the virus that you want to get the immune system working against, you add that in there, and then you inject the patient with this combination of virus and new virus. And that's what J&J did. They initially were using a virus that is most people have been exposed to. It's an adenovirus 5. But what they found was that adenovirus number 5, people basically just, because they're immune to it already, they just reject the whole thing, and it doesn't give it a good enough immune response. So what they're now using is adenovirus 26, which is a different type of adenovirus, but it enables the virus to stay around a little longer and induce the immune response somewhat better, and to COVID. The J&J vaccine, which was presented to the advisory committee on Friday, that it's a single dose as opposed to Moderna and Pfizer, which are 2 doses. And the difference between a virus-induced response and Moderna and Pfizer, I'll explain in a second, but the fact that you can get away with 1 dose and it's a virus is very easy to make and it's very easy to produce. So that should be a very stable and good virus, provided, of course, that the virus that the vaccine, and this applies both to also to Moderna and Pfizer, the virus isn't mutating away from whatever the vaccine is, again, whatever the injection is. The injection is giving a certain range of proteins, and what the virus is doing now is it's mutating away from that range. That's why you're getting immune responses against a lack of immune response against the South African virus or the UK variant. The Pfizer and Moderna made their vaccines against the original, what's called the Wuhan strain, which rapidly changed to the Italian strain, which is what infected all of New York and then the rest of the US before changing even further. But the coverage that's given looks like it's going to work for most of the strains. Whether it's going to work for all of the strains, I don't know, and neither do Moderna and Pfizer. And statements put out by them say they think it will work, but they are spending a lot of money developing booster shots against the new variants. If it's going to work against new variants, why are they spending money on booster shots? That's just something you need to think about. But J&J are using a well-established technology of putting a virus or a somewhat crippled infectious agent into a patient, produces an infection, and then the body responds to it. Moderna and Pfizer, what they have done is they've taken something called mRNA. mRNA is, everyone's heard of DNA. DNA is the genetic code of a person, but DNA can't make protein. You can't go from DNA to a protein. The way the body goes from DNA to a protein is it uses something called mRNA. So mRNA then gets transcribed by something called ribosomes within a cell to make protein. So what Moderna and Pfizer did was they took mRNA from the virus that could then be injected into patients, and then that would then get what's called transcribed into protein. That protein is expressed, and then the body makes the immune response. So they sort of circumvent the virus and let the body produce its own antigens.
Nora asked, what about Novavax? Is that, can you respond to that, Mark?
So if I remember correctly, Novavax is also an adenovirus, but I can't remember. I can't remember 100% about Novavax. Novavax. What I do know about Novavax is that they are very well positioned because they have a lot of ex-government employees, and so a lot of the funding that goes to various organizations is who you know and how well you know them. And so Novavax, prior to COVID, were almost a bankrupt company, but they had a lot of ex-government employees, and then they managed to get a lot of government funding. And that's pushed them to near the top of the pile.
So there's another question here. Is, I guess, Howard Dykeman, I thought Kovi mab was an antibody, not something which causes the body to produce antibodies, or is Kovi gene mab a gene or mRNA? You have to coat it with PG or something and store it in the freezer. And how effective are preventative measures such as vitamin D, quercetin and zinc and vitamin C, etc.?
So I don't want, so the latter point I don't want to address because I don't know what the science is on that. There are reports, but these are anecdotal reports that they work quite well. But Kovi gene mab, what that is, it's the DNA for the antibody. So it's the antibody is introduced into a patient's arm, and it's not coated with PEG. It's a proprietary formulation that allows it to actually be something called lyophilized, which means it can be stored at room temperature, and then just water is added to it, and then it gets injected. So it's a much easier way of giving the virus, giving the gene to as many people as possible, even in the absence of freezers. Pfizer, originally they had to store their product at -70, which is Antarctic type temperatures. Now they can store it at -20, which is routine kitchen freezer temperatures. And Moderna, their product is also stored at -20.
Let's see if there are any other questions here. Is that last one similar to the Israeli therapy that is inhaled? I guess that was referring to one of the therapies that.
So the inhaled therapy, as I understand it, is not an antibody, it's an antiviral drug. Ours is an antibody.
So Mark, it seems like that there's a big race going on, right?
There's always a race. There are races between companies, and there are races between companies and the virus. And at the moment, I think it's sort of neck and neck, but the virus is not stupid and is mutating, and the mutations are overcoming some of the treatments. And we know that to be the case because it's now well documented that patients have been infected twice with virus. So that means someone could have been infected once, and then the virus mutated, and then they got infected again. Whether that infection was as potent as the first infection, that's debatable. There are some reports that it's worse, some reports that it's not as bad.
So Mark, if you were a betting man, yes, and we're in this horse race, when is the race going to end, number one? I'm not going to ask you for the outcome, but obviously the outcome, we hope, please God, should be positive in the end. But do you have any sense from your worldview?
From my worldview, I think the race will probably end in about 3 years. but we will during that 3 years, it'll be a curve, and we will be catching up with the virus, and it will end up being like a routine flu. Yes, the virus is mutating, but we will always be able to outthink the virus and give everyone the vaccination or the antibody to protect.
And you say that's getting the world back to sort of
Back to normal.
A controlled situation. What about in any sense of when the US might be? I mean, will we be having Rosh Hashanah services in shul as normal this year?
So that I don't know, but a lot of that, unfortunately, is political.
Right.
When the politicians decide that they've milked this disease as far as they can, then everything will get back to normal.
So one last question. Do you recommend the Pfizer vaccine? I don't know if you would recommend.
I'm going to claim the 5th.
And if you had no reaction to the first or second shot, does that mean it was not effective?
Well, if you had no reaction to the first or the second shot, after about 2 weeks, you can always go to, I think CVS is doing an antibody test. You can then see if you've got antibodies to the virus.
Excellent. Mark, I think you've been very, very, very informative, and I appreciate your time. Rabbi Reich had to rush off to give a bar mitzvah lesson. Thank you very, very much. Fascinating, really, really fascinating. And we can all, as the rabbi said, it's all in God's hands at the end of the day anyway. All the ingredients are out there from the beginning of time. And we turn to physicians and scientists like yourself to help give us the answers. And B'ezrat Hashem, we should all be together.
Amen. That's what we try to do.
Thank you. Thank you all.
Thank you, Mark. Thank you, everyone. Bye.
Bye-bye.
We'll look forward to the next wellness program at the end of next month. Look forward to seeing you all there. Thank you very much.